Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss the extrinsic pathway of apoptosis using the receptor, toll-like receptor 3, as well as zDNA binding protein 1. In previous videos, we discussed extrinsic apoptosis with the tumor necrosis factor receptor, and then we discuss extrinsic apoptosis with the Fas receptor as well as the trail receptor. In this video, we shall focus on extrinsic apoptosis using the receptor, toll-like receptor 3 or TLR3, and then we'll look at extrinsic apoptosis with zDNA binding protein 1. Now, toll-like receptors or TLRs are pattern recognition receptors or PRRs. These are found predominantly in cells of the innate immune system. These cells include cells such as neutrophils, macrophages, dendritic cells, and natural killer cells. These TLRs, these toll-like receptors, they bind signals called PAMs. And PAM stands for pathogen-associated molecular patterns. So these are biomolecules found in pathogens. Examples of PAMs include lipopolysaccharide or LPS that is found in bacteria, lipoteichoic acid or LTA also found in some bacteria, glycophosphatidylate inositol, GPI, found in certain parasites, the bacterial flagellin protein, diacyl lipopeptides found in bacteria, and double-stranded RNA that is seen in some viruses as well as parasites. So this is how our cells of the innate immune system, such as neutrophils or macrophages, they can recognize these pathogens, these PAMs that are present on pathogens by using their toll-like receptors. So we see this PAM binding to TLR. Now, besides PAMs, these TLRs can also recognize DAMs, and these are damage-associated molecular patterns. So these are chemicals that are released by damaged cells, our own damaged cells. It's kind of interesting that it's been found that stimulation of these receptors by our nucleic acids have been seen in certain autoimmune diseases. There are about 10 different types of TLRs that we know of in humans, and they are labeled TLR1, TLR2, and so on. And these TLRs are either located on the surface of the cell. These examples, TLR 1, 2, 4, and 5. And these TLRs recognize mostly lipids and protein PAMs. And then there are TLRs that are found in the endosome. So TLR in the endosome, examples 3, 7, 8, and 9. And these recognize nucleic acid PAMs. Now, TLR is a transmembrane protein, and it has several domains. So one domain is the leucine-rich repeat or LRR ectodomain. So that would be this part here. This is the part that recognizes the PAM. In this case, this would be the part, the ectodomain that recognizes the PAM. And then there is the transmembrane domain, and that's the domain that is embedded in the membrane, in this case the plasma membrane, and in this case the endosomal membrane. And then there is the cytoplasmic toll interleukin 1 receptor domain or TIR domain. So that would be this part here or this part here for the endosomal receptor. In this video, we shall use TLR3 as an example. So this is a endosomal toll-like receptor. So this is right here in the endosomes. As we shall soon see, and we have seen this several times before, TLR3 can trigger either the pro-survival pathway where the immune cell survives but releases chemicals that stimulate inflammation, or the immune cell can die by apoptosis or by necroptosis. So let's look at these programs. As before, it starts with the ligand binding to the receptor. So in this case, the receptor is TLR3, and the LRR domain of TLR3 will bind the ligand, and the ligand in this case is double-stranded RNA. As we discussed earlier, TLR3 is a toll-like receptor found in the endosomes, and it will bind nucleic acid PAMs that are nucleic acid. So in this case, double-stranded RNA. So the story starts in the endosome of the innate immune cell. This binding of the ligand to TLR3 will cause the TIR domain, this TIR domain of TLR3, to interact with the TIR domain of this protein TRIF. So this TRIF protein is recruited to the receptor. TRIF stands for TIR domain containing adaptor-inducing interferon B protein, TRIF. Now the TRIF protein contains the RIM domain, which is RIP homotypic interaction motif, and this RIM domain, as we discussed in previous videos, is also seen in proteins such as RIPK1, RIPK3, and also the protein zDNA binding protein 1, which we'll discuss later. So the RIM domain of TRIF will interact with the RIM domains of RIPK1 and RIPK3, and that will recruit these proteins towards the receptor. The RIPK3 and RIPK1 are recruited towards the TLR3 receptor, which has TRIF bound to it. So with the help of RIPK1, several other proteins are recruited, forming this very large complex at the toll-like receptor 3. We saw a sort of a similar type of complex when we discussed extrinsic apoptosis by the tumor necrosis factor receptor in a previous video. Similar to what we saw in tumor necrosis factor receptor, this complex now causes activation of the NF-kappa B as well as the MAPK signaling pathways, and these result in increased expression of pro-inflammatory genes as well as pro-survival genes that we discussed in video 8.13 under extrinsic apoptosis with tumor necrosis factor receptor. So the cell survives and produces proteins that cause inflammation, proteins such as interleukins and chemokines and so on. Now, this complex also activates a transcription factor called IRF3, which stands for interferon regulatory factor 3. And this transcription factor will move to the nucleus, will travel to the nucleus, and it will enhance or induce the production of interferons. Now, as we shall see in future videos on the immune system, interferons are very important proteins produced by the immune system that interfere with viral infections, and they also help modulate the immune response. Now, the caspase 8 in this complex cleaves and inactivates this protein called N4BP1. This is a protein that suppresses cytokine production. So by cleaving this protein, that provides for a stronger inflammatory response by this innate immune cell. So the end result of this pathway would be the survival of the cell, which produces several inflammatory chemicals such as cytokines. Now, it's not entirely clear how the apoptosis pathway is triggered, but it seems like the deficiency of certain proteins such as LUBAC here or CIAP12 will cause the caspase 8 to be recruited by FAD, and this will then trigger the cleavage and the activation of the executioner caspases, which will cause the innate immune cell to undergo extrinsic apoptosis. Now, if caspase 8 in this complex is inhibited, for example, as we discussed in previous videos, many viruses produce inhibitors of caspase 8, inhibitors such as the protein FLIP, then what can happen is the cell undergoes necroptosis. So in this case, caspase 8 does not work. So caspase 8 actually breaks these proteins that are very important for necroptosis, RIPK1, RIPK3. And now since caspase 8 doesn't work, these proteins are not broken down. And so these proteins will recruit this very important protein MLKL that we'll talk about under necroptosis, and that will lead to necroptosis. So if the viruses that are growing inside this innate immune cell produce FLIP, and these FLIPs are inhibiting apoptosis, the cell still can die by necroptosis. So necroptosis, as we mentioned earlier, probably evolved as an antiviral defense mechanism. Okay, so now let's look at the receptor zDNA binding protein 1 or ZBP1. And this is a receptor that binds the signal, which is a Z form of RNA. Now, the receptor has two Z alpha domains, so Z alpha 1, Z alpha 2, and these domains recognize Z nucleic acid, the Z form of nucleic acid, which is made by certain viruses such as the herpes simplex 1 virus, the cytomegalovirus, as well as the influenza A virus. So besides the Z domains that bind the Z form of RNA, this ZBP1, this receptor, also has two Rim domains, Rim1 and Rim2. And these Rim domains, as we mentioned earlier, can bind proteins that also have Rim domains such as RIPK1 and RIPK3. So it's not surprising in the next step, the Rim domain of the receptor ZBP1 interacts with the Rim domain of RIPK3. So the Rim domain of the receptor interact with Rim domain of RIPK3, and that recruits RIPK3 towards the receptor. Now the Rim domain of RIPK3 as well as ZBP1 now interacts with the rim domain of RIPK1. So RIPK1 rim domain now interacts with the rim domain of RIPK3 as well as ZBP1, and that will recruit RIPK1 towards the receptor. Now, if you recall, RIPK1 has a dead domain or DD. So the dead domain of RIPK1 now will interact and recruit FADD, because FADD also has the dead domain. So this dead domain interaction will cause FADD to be recruited by RIPK1 towards the receptor. Now, as we mentioned in the previous video, FADD also has, besides the dead domain, the DD, it has also the dead effector domain or the DED. So the DED of FAD now will interact with the DED of procaspase 8, and that will recruit caspase towards the receptor by FAD. So this recruitment of caspase will result in its dimerization, autocatalysis to form the active caspase enzyme, and now this active caspase 8 enzyme will cleave the executioner caspases, which will then lead to apoptosis. So this is how the ZBP1 receptor, by binding Z form of RNA found in these viruses, can cause apoptosis in the cell. Now, once again, if for whatever reason caspase 8 is inhibited, for example by the viruses making their flip proteins or some other reason, then the RIPK3 can now recruit MLKL, and that can lead to necroptosis. Okay, so this is another possibility. If the cell cannot die by extrinsic apoptosis, it can die by necroptosis. Now, besides triggering apoptosis or necroptosis, it's also been found that the ZBP1 pathway can promote the expression of interferon-stimulated genes or ISGs. And stimulation of these genes produce several very powerful antimicrobial proteins that will help fight the infection. Okay, so this is how the TLRs and the ZBP1 receptors can lead to apoptosis. In the next video, we shall focus on this part here. We didn't talk of it. We always mention in previous videos that somehow there's activation, cleavage and activation of the executioner caspases. So now the question is, what do these executioner caspases do? How do they cause the cell to die by apoptosis? So that's what we'll do in the next video. We shall discuss the actions of these executioner caspases. So until the next video, thank you very, very much for supporting this channel, sharing this information with your colleagues, and we'll see you in the next video.